Hello everybody, this is Varsha Kulkarni. I'm from Darwin Academy. Today let's discuss something about the nucleus, which is also called as the brain of the cell. We are going to discuss a little bit about nucleus, its different parts and the functions that each part would perform. Alright, so we all know that Robert Brown discovered nucleus in the year 1831. So imagine in way back in 1831, it was Robert Brown who discovered nucleus. Alright, so if I have to study any animal or plant cell, I generally would stain it. Here, if I take cheek cells, that is the animal cell, and stain it with methylene blue, I would be able to distinguish the different parts of a cell. Let me explain this in detail. You might find the sentence very vague. Let me sort that out for you. Now, if I want to study an animal cell, okay, I would be taking a human cheek cell. How do I take this cell in the first place? I'll be taking a spoon, putting that in the mouth and scraping the inner surface of the cheek with the help of the spoon. And in that spoon, I would be getting a bit of cheek tissue. I would be placing that tissue on the glass slide and I would be adding a couple of drops of stain to stain the material, to stain the animal cell or the animal tissue. Alright. Why am I staining this? If I don't add stain, since these cells are not colored naturally, I would not be able to distinguish any part, or rather I would not be able to see any different or any part of a cell. If I place a couple of drops of this methylene blue stain, then the nucleus, the center of any particular cell, which is a dark spot, would take up more stain and the outermost boundary of any cell would take up a lighter stain. Let's go to this first diagram that I've drawn. I have drawn an animal cell as we have observed it under a microscope, under low power. Alright, if I would simplify that sentence for you. When I've placed the cheek cell on the microscope slide, stained it appropriately, then after that, if I observe that slide under the microscope, under low power, then I would be able to see many such small little round structures. Remember, I have magnified the image here millions of times. Cells are very, very tiny. Alright. If I observe that tissue, cheek tissue under the microscope, then I'll be seeing many such cells one beside the other like this. Alright. And I would be seeing something which is a dark spot probably at the center because I have stained it. It would be dark blue in color. That region is actually the nucleus. Alright? And the boundary of any particular cell would have taken a lesser lighter color when compared to the nucleus. The boundary of a cell is the cell membrane. And in between the cell membrane and the nucleus, this region where I've shown the dotted structure or where I've marked it with the help of dots, that is the cytoplasm. Remember, cytoplasm is a jelly-like fluid which is present within the cell. Let me reiterate what I have discussed up until now. We are looking at the nucleus, which is called as the brain of the cell. Now, who discovered nucleus? It was Robert Brown who discovered nucleus in the year 1831. If I observe any tissue under the microscope, I would rather see it this way, right? You know that tissue is a group of cells which have the same structure and perform the same function, isn't it? Now, if I focus or if I magnify that single cell in particular, then under the low power in a microscope, I would be able to observe only 3 different parts. Remember, I should have stained the material appropriately. What are those three different parts? The first part that I'm going to see, that is the boundary of any cell, which would have taken up a lighter color when compared to the nucleus. Right? The lighter outer boundary which you see in a cell is the cell membrane. Then the darker spot that you would have seen, right, that is the nucleus. And the space between the cell membrane and the nucleus would be somewhat lightly stained, and this is the liquid which is called, or the fluid which is called as the cytoplasm. I hope this is clear to all of you. With that, I'll be moving on to the actual details of the nucleus. If I focus this nucleus under an electron microscope, rather, if I use an electron microscope to study the different parts of a nucleus, this is what I'm going to see. Alright. So I have also magnified that image millions of times. Right. Cell is very tiny, and imagine if I'm talking about the size of a cell, think of the nib of this particular pen. It is very tiny, isn't it? This nib of this marker pen would accommodate millions of cells. Such being the case, think of the size of the nucleus which is within every cell. So it would be very minute. And if I observe that under an electron microscope, I would be seeing the different parts. Hope you have understood what is the size of a cell and also what is the size of a nucleus. Alright. Now, let us study the parts of the nucleus in much more detail. Just like the cell having the cell membrane, the nucleus is also covered on the outside. The nucleus is covered on the outside by a nuclear membrane. The speciality of this nuclear membrane is it is two-layered, whereas the cell membrane, we are not talking about the layers for grade 9. Alright, this is generally single layered for your understanding. If you go to higher grades, we will learn that the cell membrane is made up of, again, two layers of lipids, and there is protein also. Okay. That being the case, I don't want you to get confused. Let me go back to nuclear membrane being double layered. Okay. Then its composition, or the components that make up the nuclear membrane, is almost same as that of the cell membrane. And there is another feature which I would like to tell here. This nuclear membrane is not continuous. It is interrupted at every regular interval by a hole or a pore. And that pore is called as the nuclear pore. Okay. The nuclear membrane's job is to protect the contents of the nucleus, and also to see what goes in and what goes out and what comes into the nucleus. Okay. Then what should be the role of the nuclear pore? Nuclear pore allows only specific substances to go in or go out of the nucleus. So it literally acts as a passage for the movement of materials into and out of the nucleus. Okay, I hope the structure of the nuclear membrane and the nuclear pore is clear for all of you now. Moving on to the next part, the nucleoplasm. Just like the cytoplasm, there is fluid within the nucleus which is called as the nucleoplasm. Okay, this is also a fluid. It also has a couple of enzymes just like the cytoplasm. Then we see a loosely intertwined thread which I have drawn in the green using a green marker, and I have labeled it as chromatin material. What are these chromatin materials? Chromatin material is actually DNA and protein. But during the cell division, the chromatin material condenses to form the chromosomes. Alright, did you understand? The chromosomes are only seen during the cell division. How did they appear all of a sudden during the cell division? It is from the chromatin material, or rather, the chromatin material has condensed a lot to form the chromosomes only during the cell division. Otherwise, the chromatin material exists as loosely kept thread within the nucleus. This chromatin material is made of DNA and proteins, okay? And the specific regions of the chromatin material, or the chromatin material later on condenses to form the chromosomes. And on the chromosomes, there are specific regions that are called as the genes. Genes, not the jeans trousers that you wear. These genes are those regions on the chromosomes that have characters which help us inherit those traits from our parents. Or rather, the chromosomes have genes that are the hereditary or they help us, or these genes carry the characters from parents to their offspring. Alright? Offspring is the correct word. There is no word called as offsprings in English. Alright? Am I clear? Anyway, let me get back to all these once again before I wind up the session. There is another part within the nucleus, which is the nucleolus. I have used a blue colored marker and it appears like a round structure. Okay? The nucleolus, the main job of this is to make the subunits that actually help in the protein synthesis. For the protein synthesis, we would require ribosomes. So, ribosomes are being produced by the nucleolus. Alright, I would like to repeat whatever I have discussed. If a nucleus is magnified many times, or if I study a nucleus under electron microscope, I would be able to see a structure which is somewhat similar to this. Right? Remember, I have drawn this diagram. The diagram is huge because I would want you to appreciate each and every different part and also understand its functioning. It will be way too tiny in real life. The nuclear membrane, I'm repeating, please be careful. The nuclear membrane is a double layered membrane which is the outer covering of a nucleus. It is discontinuous, that means it is not continuous and is interrupted by nuclear pores. Okay? The nuclear pore allows the transport of substances into and out of the nucleus. Okay? There is nucleoplasm which is similar to cytoplasm in its composition, and it is fluid or liquid like in nature. The nuclear membrane structure is also similar, it is also similar to the cell membrane. The chromatin material is actually the genetic material. Okay? This carries the information that the parent would be giving to their child. That is a simplified version of telling that it is the genetic material. So, the chromatin material is a genetic material which carries information from parents to offspring. Alright? For example, I would have inherited a few characters from both my parents. How did I get it? It is because of the chromatin material which has specific regions called as the gene that I would have inherited from my parents. Okay? So, the chromatin material is a loosely wound thread. Okay? It is made up of DNA and proteins. Alright? And during cell division only, the chromatin material condenses to form the chromosomes. So, we are going to see chromosomes only during the cell division. The nucleolus is the last part of the nucleus which I will be discussing. The nucleolus is a round structure. It helps to make ribosomes. Ribosomes are later used to synthesize proteins. We would be talking more about protein synthesis and how these ribosomes are used when I'm talking to you about the endoplasmic reticulum or the ER. Okay, so nucleolus, as of now, you should know that it helps to make the subunits of ribosomes, which are later utilized for protein synthesis. I hope with this, the concept of nucleus is clear for all of you. Nucleus the cell under a resolution or low power of a microscope would appear somewhat like this. But if I focus on the nucleus under high power, or if I observe that under an electron microscope, I would be able to see all these parts. So with this, we would conclude the topic of nucleus. Thank you.